Welcome back, ladies and gentlemen. So we're going to continue our discussion on sort of population dynamics by now talking about population density. So when we talk about the density of anything, we're talking about the measurement of something per a given unit of area or unit of volume. In the case of population density, I want to know the number of individuals within that given space that I'm studying. Because just having a simple population size can only tell you so much. How tightly packed are they within that particular space that they are given gives you a much bigger understanding of how that population is going to interact with each other, and then continue to interact with their environment as well, when you know how densely populated that area is. So if we put that again into some sort of math terms here, D sub P is your density or population density, N is your number of individuals, and then that is divided by a given unit of space. And that unit of space can be acres, it can be hectares, it could be um, square meters, depending on what you're talking about in some sort of terrestrial environment. Or you could talk about cubic centimeters or cubic yards, whatever the case might be, milliliters, gallons, if you're talking about something that is more of a unit of volume because it's aquatic instead. So when we look at population density, if we just look at humans in the US, hopefully this kind of makes sense, right? Where is the population most dense? Where are there largest numbers of people in the smallest unit of area? Around big cities, right? So I look at a lot of the East Coast, and we are very population dense here, around Los Angeles, San Francisco, Seattle. And then you sort of go out in the middle of the country, in the middle of farm fields in Kansas or Wyoming, and you may not see people for miles and miles and miles and miles and miles. Um, People are much more spread out there. They're not living on top of each other. And even if you use the example of here, right, much more densely populated here in town, you spread out into sort of the more rural areas around here, it spreads out even further and go down into the city and it's even more densely populated. So you've got a lot more people living in a much smaller space, and that does have impacts for how resources need to be utilized and allocated, um, competition for those resources, if we're talking about um, organisms who compete over certain food sources or space, all of that does matter. So, how do we figure this out? Well, when we're doing ecological studies, we have a handful of techniques that can help, and this is just a very small snippet of some of those potential sampling techniques. So one thing we can look at are creating coordinates, all right? So we mark off a given area, call it a coordinate, and within that given space, we count the number of species of interest within that quadrant. So sometimes you'll see these little like PVC squares. Those can be used, or you may see them sort of come out with twine and sort of make a grid pattern. And then this quadrant sampling is repeated for several locations within your given study area. The average results then are estimated together based on the number of individuals that you count, as well as the amount of space that you have counted within, how many quadrants did you count, and how big are those quadrants, to sort of estimate the population density within the ecosystem. It is an estimate. You are not going to be able to count every organism. You're guaranteed to miss some. And this quadrant sampling is really useful for things like plants, because those plants don't get up and move on you. But if you're talking about insects or birds or any organism that is capable of getting up and moving, that is going to have to be taken into consideration when you're making these estimates because those things can move. So it's possible for you to have never counted a subset of the population because they just never ended up in your quadrants, or it's also possible for you to have counted a particular individual more than once if they moved from one quadrant to the next when you moved in order to count. So there are definitely benefits and drawbacks to all of these different counting methods. And in this case, probably more acceptable, more reasonable to utilize when you're talking about plant species, also when you're not talking about a huge amount of area to cover, because this is going to take time. Another option is this idea called indirect counting, and this is much more effective for organisms who do in fact move around, or who are organisms that just tend to be very difficult to see or interact with as a human. They're skittish, they're not going to come near you, um, whatever the case might be. So in indirect counting, you're not counting the actual organisms, but instead you're counting things that they would create. So nests or burrows or tracks, rather than the organisms themselves. Again, not going to be a perfect system because you may be counting tracks that are left by the same organism more than once, right? Um, and so you really do also need to have some basic understanding of the organism that you're studying in order to do this. What do their nests look like? What do their burrows look like? Um, you don't know how long that nest has been sitting there, right? It could be an empty nest that was just never got destroyed from 3 years ago, and that organism is long gone. So again, benefits and drawbacks, right? Helpful when you can't directly count those individuals because you can't get to them, you can't see them, whatever the case is. But definitely potential flaws in the type of data that you collect. Mark recapture is another option, and this one's kind of interesting. We do this one actually a lot for organisms that tend to move around. Also, a big one when we talk about migratory organisms. So the idea here is that we trap the animals in our study area and mark them in some way. Colored tags, colored dyes. You can do something as simple as a little bit of like nail polish on an organism if it's not going to be something that's going to be exposed to the elements for too long. We do a lot with GPS and radio tracking tags now. And the idea is when you trap a subset of those individuals within your population and you mark them in some way, you're then going to turn around and release those individuals back out into the wild. After a given period of time, you then recapture the same number of organisms and count the individuals that have that marking, whatever it might be, the tag, um, the colored dye, whatever, as opposed to the number of unmarked individuals. And then you do some simple math here. So we estimate the total population size to be the number of individuals in that first capture that we initially tagged times the number in the second capture total, right? So if I capture 100 here and I capture 120 here, then I'm going to multiply those numbers together. And then I'm going to divide that by the number of organisms in that second capture that were marked from the first one. So if I marked 100 individuals, I captured 120 the second time, I'm going to multiply those numbers together. And of the 100 that I marked the first time, I get 75 back. That would be the number down here. And that's going to give us a rough estimate of the population size. Again, really nice when we talk about things like migratory organisms, how many of them make it down, how many of them make it back, and what does that mean in terms of their survival rates, how they migrate, what's happening to them during that migration pattern, and are they successfully making it back to the same location, and is that just how they work? Do they always come back to the same place? So when we look at overall factors that affect density, it really is going to be what's affecting the population size within the area that we're studying. So we've already talked about population size from just the standpoint of natality or that birth rate, as well as the mortality or the death rate. So births are clearly going to bring in new individuals and increase the population. Mortality is going to remove them. But if this is an organism that is capable of moving, so it's not a plant, it's not something that stays stagnant, then we can also take into consideration things like immigration and emigration that will also affect the population size and in turn affect the population's density. So when we talk about immigration, we're talking about individuals moving into a population. More immigration is going to increase your population size as opposed to emigration, where the individuals are moving out of your given population within that given area, and so they will end up decreasing that population size. And so it really is the overall play of all four of these particular events that then determines the natural rate of increase for your population. And then if we constrain that based on a particular study sample space area, then we can talk about how that ends up affecting the population density as well. So again, natality or birth rate is going to increase your population. Immigration is individuals moving in, so that will also increase your population. And then the mortality and the emigration, both of these will end up decreasing the population size, so death rate as opposed to individuals moving out. And it's the overall net of all four of those processes happening within a natural population that determines, is that population growing overall? Do you have more birth rates and or immigration happening? Or is the population shrinking because you're having more deaths and or more individuals leaving via emigration?